Alright, so let's move on to new approaches to gene therapy. Now that you've known the basic, you have seen some of the scope, application, and the possible effect. Let's have a more detailed. So if you remember, in the second video, I did mention about a dominant negative mutation because, like I said, among the 5 criteria, sometimes it doesn't really matter if the gene involved is a dominant negative mutation gene, then really basically there's nothing you could do about it. Why? Because dominant negative mutation is caused by mutated genes that prevent the normal function of the original protein. So even if you add a normal gene back, it will not help because it will still be dominant, it will not turn it back on, or simply said, it will be lost forever. However, one could basically try to either repair or eliminate the mutated gene, and basically there are a few approaches which we'll see in a while. So to treat those kind of situation, specific and efficient means of delivering the gene to the target cells is required. So I have a video about the negative and also dominant negative mutation that you can view. I will put the link in e-learning for you to see. So now, oh, so sorry because I think I stretched this picture so it's a bit blurry, but you can find it in Google if you just Google up what is dominant negative mutation. So basically what happens is that, okay, when a gene is mutated, for example, there is a mutation there, so it wouldn't be able to be transcribed, and of course no transcription means no translated protein, so protein couldn't be made. But in a case whereby it still made it to the translation, so that means that the protein is actually a mutated protein. So as compared to the normal protein, as you can see over here, when they interact, so it will basically block its function. It will interact because there is a pocket, okay, there is a binding site, so it will interact, okay, but the interaction will prevent the protein from functioning or from performing its function when it is combined together, and that is not good about dominant negative mutation. Apart from not being able to ensure the normal function of a gene, it basically blocks the function of the gene as well. So one such technique to repair the mutation is called the SMART technology. If you see this sign, and we will discuss this in detail in chapter 6 on intellectual property, this means that it has been trademarked. So the process is basically just unique to them, and well, if anyone else wants to use it or want to buy the technology, they have to pay them a sum of money. So now SMART, what it is a short form of spliceosome mediated RNA transplicing. Again, I'm not going to explain what spliceosome is. Okay, try to remember or Google up if you have forgotten. So this SMART technology is to target and repair mRNA transcript copied from mutated gene, because regardless, most of the time, sometimes it still made it to the transcription and also to be translated, but then protein will not be able to be function properly. So the technique repairs only the section of mRNA transcript containing the mutation instead of attempting to replace the entire gene, because by right, generally that couldn't be done. So once the gene is copied into the mRNA, the RNA-based machinery called spliceosome is used to cut out the non-coding introns and join the exons together. So these are the steps, and you can view it in the picture, again, in the next slide or in your notes. So deliver the RNA strand that pairs perfectly with the intron next to the mutated RNA segment that prevents spliceosome from including the mutated segment in the final splice product, to correct the version of segment whereby it is delivered simultaneously to replace the mutated segment, and by doing that, normal function protein could be translated from the repaired mRNA to ensure that the protein could function properly later on. So this is the case of normal gene splicing, just to refresh back your mind. So remember the transcription process, so whereby when you have an exon and intron, and this only applies to eukaryote, so you need to cut off the intron, and the intron is cut out by the spliceosome. So spliceosome cut, okay, at both ends of the introns, and later, okay, your exon is joined by the ligase, so to form mature mRNA before proceeding to translation. But again, using a SMART technology for gene therapy, this is what happens. So they found that, okay, there is a mutated version of the gene in one of the exon, for example, exon 2, as you can see over here. So what happens is that, okay, they create a basically exon part 2, okay, without the mutation, the correct version of the gene to be included or to be replaced, and this is what happens. So the engineered mRNA strands, okay, will bind to the intron because it has a binding domain. This binding domain basically is complementary to the intron, so it will bind to the intron over here, okay? And then next The spliceosome, will assemble on the mutated version of the exon 2 and on the engineered RNA strand. So it will stay here and here. So the spliceosome will later on remove the intron, along with the mutated version of the exon 2. So it will cut here and also here. So how can you make sure that it will cut here? It is actually a bit tricky process, but you can actually design a basic spliceosome, to make sure that it cuts, at this part and not, this part, so that later on it would be of no use if it were to cut over here. It has to cut here and also here towards the end. So what happens that once it has been removed, the one that is defective, the exon 2, without the mutation, the ones that you manufacture in the lab or you modify, could now be joined to the first exon. So now your exon 2 is without the mutation because it has been modified. So when it basically produced the mature mRNA and later translated to protein, the protein could function and basically should be able to work normally. Now, apart from that, there are a few other techniques. So SMART technology is being used mostly for dominant negative mutation. Some with a success, some not so much of a success, but it has been proven useful, even though probably not high percentage of success. So let's take a look at some of other techniques to prevent mutated protein. So I'm highlighting 3 here. There are a few more, but these are the most common ones. So the first one is triple helix forming oligonucleotides. So if you remember your DNA basics, you only have double helix, right? And even for protein structure, you only have up until double helix. So what happens is that, when triple helix, it means that it will perform triple helix instead of double helix. So it will target mutated gene DNA sequence to prevent the transcription. So you can just read on your notes. I'm going to explain again by this diagram. So what happens is that, this is your gene, double-stranded, remember? So your mutation is on one of the side of the strand, so either sense or antisense. So you include another oligonucleotide. So oligonucleotide is just a short strand, similar to your primer as well. So your primer is also considered as oligonucleotide. So your oligonucleotide is the one that is complementary, or basically similar to the one with the mutated version. So what happens is that, when you add in the oligonucleotide, it will form triple helix over here. So you have 1, 2, 3 strands, so triple helix. So what happens is that in this case, when triple helix forms, it will prevent transcription. So no transcription, no protein will be made, no protein translation. So prevention at the very beginning, but that also means that probably the gene will not be expressed or will not be made. Second technology is on antisense. So the target is basically to target mRNA transcripts copied from mutated gene to make sure that it is turned off. So as gene contains double-stranded DNA, of course, the sequence of one strand transcribed to produce mRNA during transcription is a sense, and the opposite is the antisense. So this is what I want you to find out previously when I said on the antisense strand. So antisense is the antisense RNA, is delivered and binds to mutated sense RNA strand to prevent translation, and it involves 2 steps, whereby the antisense RNA strand coding for the mutated gene is being delivered and bind to the sense strand RNA. So better basically look at this picture as I explained. So you have your mutated gene similar to the first scenario, only on one side. So you just need to make sure which side it is basically mutated on. So whether it's on the sense or antisense. If it's on the antisense, then we wouldn't call it as sense technology. There is another way, or you just use triple helix. But if it is on the sense strand, so you could make another antisense, a new mRNA sense strand to bind to your sense strand. So what happens is that the antisense RNA strand that binds to your mRNA strand will prevent translation. So product could still be made over here, but it will prevent protein from being made and from producing dysfunctional or non-functional protein. So the third one is by using ribozymes. So as the name says, so it is an enzyme, and ribo, which means that it is to cut. So it has similar aim to antisense to turn off the mutated gene transcript from being translated to prevent further damage to the system and to the regulation. But RNA molecules, okay, will act as an enzyme, which is the ribosomes as an enzyme, as a molecular scissor, similar to your cutting mechanism in the RNA, sorry, the SMART technology just now. All right, kind of like spliceosome. So it involves specific binding of ribozyme RNA to the mRNA of mutated gene, where the target RNA will be spliced to prevent protein translation. So it involves these 3 steps. So delivery of RNA strands engineered to function as ribozyme, specific binding, and cleavage. So as you can see over here. So first, identify the mutated site of the gene. So strand separation. So if it is on the sense strand that will be made again into mRNA, so mRNA has already been transcribed. So what happens is that another ribosome is being added. So the ribosome has a machinery to be cut. So it will bind, and of course it has to be complementary to the mRNA that is basically contained in the mutated site. So when it binds, what happens is that, okay, because it has a binding site, okay, so your spliceosome, okay, or ribosome, okay, cleave mRNA strand, preventing the translation. So there is a specific nucleotide sequence that signals for it to be cut. So it will cut there, cut it off. So that means that this side, okay, will be left out. What happens is that it may still be translated, but most of the time it will not be translated, seems because it's going to be truncated. So from the 3 technology, simply said, the summary is that it prevents either transcription, but definitely it will prevent translation from to prevent protein from being made, okay, that will be dysfunctional, non-functional, or blocking the original function of interaction with another protein. All right, so now for this week I only have, I think, 1 activity. I actually couldn't remember, but I think only 1 activity. So activity 3 is for you to do either individually, or you can discuss with your friends by WhatsApp, by calling, whatever means. All right, so first and foremost, let's take a vote. I have opened up the link in your e-learning, so just go there and answer yes or no. Can gene mutation be prevented? Okay, so there's only 2 answer or 2 choice. You answer either yes or no. There is no or. Now, depending on your answer, regardless whether you say yes or no, okay, if your answer is yes, how can it be prevented? If your answer is no, it couldn't be prevented, why could it be prevented? So you need to be prepared. If you chose yes, okay, you need to be prepared on how it could be prevented. If you choose no, why couldn't it be prevented? So this is what you need to do. Okay, you can either, like I said, okay, discuss among your friends, or find out more information, okay, for yourself to support your answer. So vote your answer in the link. Basically just prepare, okay, your answer for this, and we will discuss it on Tuesday later on. Okay, so that is for you to do. So I will just sum up video 3. We have another video, I think the last video 4. So to sum up on video 3, I have the delivery mechanism of your basically transgene for gene therapy. So how it is being delivered, so the explanation is actually over here, okay, you can just refer to it, okay, but okay, having or taking a look, okay, at this diagram, this is what you need to do, of course, beforehand in the lab. Okay, so you need to develop, you need to manipulate, whatever that you need to do, okay, develop your DNA-based therapeutic agent with suitable vector, either viral vector or non-viral vector, which we will discuss in the next video. So this DNA-based therapeutic vector complex, okay, with your vector, is being basically delivered to the target site. So it will first and foremost, okay, bind to cell membrane first, okay, because when we talk about eukaryote, okay, there is a transmembrane kind of like restriction. It couldn't just enter into the cytoplasm just like that. It has to have kind of like a permission, or basically kind of like a border agency that allows it to be, or to enter into the cytoplasm. So what happens is that the vector, especially if it is a virus, okay, it will bind to the cell membrane, or being on the surface of the cell membrane, it will create a hole or entry mechanism into the cytoplasm. So once it enters into the cytoplasm, it will create an endosome. Okay, so this is kind of like just a small place for it to just stand by in the cytoplasm. So next thing is that when everything is okay, and you have to remember this is still a foreign material, right, into a cell. So it needs to be very careful. It needs to be wary of the immune system, because immune system will recognize this as a foreign and it will attack it. So the function of endosome is to ensure that your therapeutic agent is safe in the cytoplasm before it is being delivered into the nucleus. So once everything is okay, so basically it will create endocytosis. So your therapeutic agent will be released from the endosome. It will be in the cytoplasm. So this is also where they need to be very careful. Okay, they need to basically make sure that they are safe from the immune system. So the next step, okay, is for the therapeutic agent to be released from the vector. Up until now, the vector is still holding your DNA. So the vector has to release your DNA, package it. So if you use viral vector, it has a good packaging system. So it will package and release your therapeutic agent. Basically mingling around, okay, in the cytoplasm. So from here, it has to find its way. It's actually a very complex mechanism, so I'm not going to put those details here. We'll just basically learn on the basic. So it will just make its way to the nucleus of the host or the cell or the new place or the patient, and basically it has to make its way into the nucleus to make sure that it is integrated and basically divided or dividing through the mitosis and the meiosis as the cell divides. So what happens if it just goes into the nucleus but doesn't integrate and replicate along? It could still be expressed, but probably it may not be very good, and you have to deliver and deliver again the therapeutic agent. This is what I meant early on, okay, in the first two videos, saying that again you need to do subsequent or follow-up therapy for this purpose. So the explanation is actually here. Okay, so you can just look on it. What I've explained is actually just a simplified version. This is a very detailed version. Okay, so I'll just leave you guys with this, and in the fourth video we will see, I think I'm only including the viral vectors, so I'll leave the non-viral vectors I think probably for next week. So let's just move on to the next video.